put my pronouns just in case. Oh, yeah.
All right. Sounds good. Well, welcome everyone to the Virtual Insect Fair. Today we'll be talking about insect damage, and I'm really excited. We have an excellent group of panelists here who have done a lot of really exciting work within this field, and I'll just turn the time over to them right away so they can introduce themselves and you can get to know them.
Alrighty, I will go first. Present. As Sam just said, welcome to Insect Damage. My name is Tessa, and I'm an entomologist. I do research outside in Riverside County, as well as working in greenhouses and in my lab. My overall research is on plant, virus, insect vector interactions. And here I am outside in the field. So I study in the field wild squash and nightshades in Southern California. These plants might be familiar to you. The one on the far left with big white flowers is related to tomatoes and potatoes, whereas the center squash and the one in the far right that's growing on cactus, these are wild squash, so they're related to pumpkin and zucchini. I also study the viruses that infect these plants. So just like humans, plants can be infected with viruses. And these viruses can change the plant colors as well as make the fruit that we eat less than ideal, so more bitter or weird looking. And our agriculture and our crop plants are actually hotspots for virus production. And those viruses spill over into wild plants, like the wild squash and nightshades that I study. So in this slide, I have some plant viruses, as well as some damaged fruit and some yellowing leaves. So these viruses are spread around by insects. Aphids, whiteflies, thrips, and leafhoppers, among others, transmit these viruses by feeding on infected plants. And then they deposit viruses onto healthy plants. So they're kind of like plant mosquitoes. They fly around, pick up virus, then spit it out in the next plant. So my main research goals while a grad student is to find out what are the major crop viruses that infect wild plants, and do plants that are infected have negative consequences? Are they hurt by these viruses? And do the insect vectors that transmit these viruses, do they do better on healthy or infected plants? So one of my main experiments to find out what's going on here is called a feeding experiment using something called electrical penetration graphing technique, EPG for short. So what I'm doing is I'm creating a circuit between the aphid and the plant, and then I record the waveforms of feeding. So I'm using some physics to follow the voltage up and down and how those waves change. And I'm comparing how melon aphids, the vector, feed on virus-infected plants and healthy plants over 8 hours. So this is what that looks like. Here are some aphids, the melon aphid in particular, and in that hand, so holding onto the little wire, that's an aphid at the tip of the wire. So I glue the aphid to do my experiment. And then I hook it up over here in this, the probe connection, and I let it feed on the plant. So here's what feeding waveforms look like for probe and go transmission. So the aphid mouth parts, this is what they look like moving inside the plant. And when they taste, the voltage drops. And here they salivate. We actually can tell when they're drooling into the plant. And then here, this is them slurping up the cell contents. So this is when they would pick up virus. So I don't have any results for you yet today, but I want you all to think about what will happen. Will the aphids do better feeding on a virus-infected plant or a healthy plant? So later on, maybe we can talk about that during the panel. So insect damage can be direct from feeding. In the case of aphids, they can drain plants of their sap like vampires. But there can also be indirect damage, like carrying pathogens around. So that's all about me.
All right, so I think that I'm next. Just let me adjust what I can see here. Okay. Hi, I'm Jamie, and I'm a PhD candidate in entomology at UC Riverside. I'm actually working in the same lab with Tessa, who just spoke. And I study a very similar group of insects to the ones that she studies, but the insects that I study are called psyllids. So this is me out in the field with my best field assistant, my dog, Bindi, and this right here is a picture of a psyllid. Psyllids, that's a silent P, are very tiny insects, as you can see here. And just like Tessa's study organisms, psyllids suck sap out of plants. And they use these special straw-like mouth parts that I've circled here, sticking out below psyllid's thorax, to suck sap out of plants. And the main reason that psyllids are considered bad is because while sucking sap out of plants, they can transmit bacteria that make plants very sick. So again, this is very similar to the system that Tessa studies, but instead of transmitting viruses, psyllids transmit bacterial pathogens. How this works is that bacteria can live inside of psyllids, and when a psyllid begins feeding on a plant, it salivates into the plant, and when it does so, bacteria can enter the plant along with the saliva, where they can then multiply and spread throughout the plant, making it very sick or potentially killing it. So in California, we have a couple of psyllid species that we know are spreading diseases in this way to our crop plants that we use for food. One example is the Asian citrus psyllid, which Tobias is going to talk a little bit more about after me. And another example is the potato psyllid, which transmits a bacterial disease that can cause malformation of fruit and tubers or kill plants like potato, tomato, and bell pepper. However, we also have over 100 other psyllid species in California that, in addition to feeding on crop plants, feed on the wild plants that we strive to conserve in our areas of natural habitat. And here's just a small sampling of them. They're actually really pretty and come in all different shapes and sizes. But we don't know whether any of these other psyllids are transmitting diseases to wild plants that might harm these wild plants. So for my research, I get to visit a lot of really beautiful conservation areas around Southern California and collect psyllids and host plant samples. And to do this, I use a combination of different kinds of traps. Here in picture A is a special trap that can trap psyllids directly into a liquid preservative. And picture B is just a yellow sticky card, very similar to like yellow sticky tape you might hang up for flies in your house. And then in picture C, you can see me using what's called a beat sheet and an aspirator to shake psyllids off of plants and then suck them up without squishing them into little vials to bring back to the lab. So once I bring my psyllids and my plant samples back to the lab, I have to test whether they're infected with plant pathogenic bacteria by isolating DNA and amplifying this DNA. And the way that I can see whether the plants or psyllids are infected with pathogenic bacteria is to amplify DNA and then run it through an agarose gel, which is what I have pictured here. And these fluorescing bands are actually the DNA from the bacteria. So that's kind of cool. And finally, I also am trying to do experiments to study the effects of these psyllid transmitted pathogens on wild plants. So right now I'm doing a lot of work with the potato psyllid, which in addition to feeding on potatoes and tomatoes, feeds on native nightshade plants such as the bluish nightshade pictured here. And as I recently discovered, also transmits bacterial pathogens to these native nightshade plants. So right now I'm trying to find out whether these bacterial pathogens are having negative effects on these native nightshade plants. All right, so I will now hand it over to Tobias to talk a little bit more about the Asian citrus psyllid.
Cool, thanks for that. My name's Tobias Moyner. I'm also a graduate student with UCR, and I'm going to talk a little bit about, kind of following with what Tessa and Jamie were talking about, of a certain psyllid that affects citrus, and it's a real threat in Southern California right now. I'm going to try to share my screen, which might help, and I'm also going to set a timer because there's a lot to talk about, and I need to limit what I say. So my name is not Beth Grafton Cardwell. However, she was my boss. She's now retired a couple of years ago. And I was on this project, and we were trying to figure out the distribution and how to control this psyllid throughout Southern California. And California is very diverse in its ecosystem. So we were trying to understand how the natural climate affects population densities and how management can control this pest in different regions. So quickly, there's the pest, which is on the left, which is the psyllid. And then on the right, this is the bacterial disease that can be transmitted within the gut of the psyllid. And it is passed from the psyllid, mostly through the adult into the tree. And within 3 to 5 years, that tree will ultimately die. And if other psyllids feed upon this tree that has this bacteria inside of it, they will pick up that bacteria and it will just continue to spread. And like Jamie said, I mean, these insects are pretty small, but when you know how to look for them and where to look for them, it gets pretty easy. So here's a picture. They have 5 intermediate stages. So on the picture down here, I don't know if you can see my cursor, you have the egg, which is going to be teardropped and it's going to be yellow. And then as they molt, they're just going to be larger versions of the smaller stage before. There's some more pictures. They like to congregate on the underside of a leaf. And the adults can feed on mature and immature leaf tissue. However, the eggs and the instar nymphs, which are these smaller guys, they can only feed and penetrate the tissue of the larger leaf tissue. So it's important when you're looking for those that you can kind of isolate your search radius to those newer leaf parts. They'll produce this waxy exudate, which is important later on because I'll touch base a little bit on how ants will actually tend this and form a mutualistic relationship, which totally gets in the way of biological control that is used to control this population. So my job with Beth Grafton Cardwell was to search certain groves and look for this pest, and this was basically every day throughout the year for a couple of years to figure out where it likes to live and how that population changed through time. So I kind of became a bit of like an FBI agent, so to speak, where I'm looking for this pest, and I'm also looking for the presence of this pest even when it's not here. And this picture shows a great example. This leaf on the right was fed on most likely by an adult psyllid, and the toxin that's injected will actually cause this asymmetrical notching on the tip of the leaf. So if you see leaves that look like this, I mean, it usually means an insect was feeding on it at one point, and in this case, it was a psyllid. And that's important to know because even if the psyllid insect is not there, the disease could still be in that tree. And the psyllid does attack other varieties or closely related species of citrus. Just to note that citrus is the big aspect in California because not only is it big in commercial agriculture, it's also in like 60% of people's backyards. So this insect doesn't have to fly really far to spread this disease. It can actually reside inside of probably your backyard and you don't even know. And then the population builds and it flies into a commercial grove. And it's a problem for farmers their job and livelihood is citrus. Let's see. So Florida has a problem because there's other ornamental varieties of plants that these pests can live on and feed on, but it's not as big of a deal here in California. So why we're worried about the psyllid does feed on the tree and it does cause some overall health decline or damage to the tree. Like Tessa and Jamie said, they'll feed on the sugary juices of the tree, which harms the tree. But more importantly is that it's a possible vector for HLB, or citrus greening disease. And Hablong Bing, it means in Chinese yellow shoot disease. There's currently no known cure for this, and once a plant is infected, it will die within 3 to 5 years. By and large, there's some exceptions to this, but it's a pretty big deal. It demolished Florida's agricultural industry, and it's moving into. It's here in California. It's here in Riverside. I don't think the disease has yet found commercial agriculture citrus. It's mostly in backyards, but it's only a matter of time, unfortunately. And to find the disease, so we went over a little bit of how to find the insect. The disease, it causes this yellowing formation of the leaves. And the bottom here is a nutrient deficiency. And you can tell based on the mirrored image of the vein pattern on the right versus the left. And up here, it's going to be more blotchy. So it's not going to be a symmetrical yellowing formation HLB, and that's pretty characteristic to that. Here's another example of what it could look like. And the fruit's going to be small. The overall production of the tree will diminish, and the flavor of the fruit will also drop, and then the tree will ultimately die. And because I'm basically out of time, there is just one more thing I wanted to point out. There's this website here that's currently authored by Dr. Monique Rivera and Dr. Beth Grafton-Caldwell, and the GIS mapping is done by Robert Johnson. And this is a really cool website, and it's filled with resources. If you're a commercial grower, you can click on this link, and it'll give you an interactive map of where the psyllid is in relation to your farm. It'll also give you management strategies based on the region that you live. It has everything from how to best monitor the pest, what insecticides are known to work in that area with your particular climate, what insecticides to use at certain times, and then other general, there's a lot of regulation that goes into HLB management within California. So this is really important for commercial. And if you're a homeowner and you have citrus, the same thing exists. Oh, okay, it's freaking out. We'll go somewhere else. And this is that map that I was referring to earlier. You can actually type in your address into this, and it'll give you where HLB has found in relation to your home. And that's important to know for a couple things. And this is regularly updated, by the way. And one reason why that's important is based on the location of where your house or your protected citrus trees are in relation to that HLB positive tree or psyllid. There's different scenarios, and this is by Citrus Insider, which is also a great resource. And this can tell you what actions you can take based on where you are in relation to that hotspot. And it is important that homeowners take action so they don't negatively affect the livelihoods of the citrus farmers that have been in California for a very long time. And with that, I'm over my 5-minute mark, so I'm going to stop talking. Thank you.
All right, guess it's me. Share with this one. We'll go into presentation. All right, guys. My name is Dina Hussain, and I am also a PhD candidate at UCR. However, unlike my colleagues earlier who spoke, I study the behavior and biology of natural enemies of insects that are labeled as pests. Now, my main focus is on the natural enemies part. because they can be utilized in a biological control program to suppress populations of an invasive pest. Now, when I say an invasive pest, what I mean is that it's not native from around here. It probably hitchhiked a ride, maybe on a boat, maybe underneath your shoe, who knows, from a different country. But the point is, this is not its native region. So I try to look at its native region to find these natural enemies and bring them over so I can apply them in a biological control program. Now, my pest that I'm studying falls under the group of ambrosia beetles. You might ask yourself, what are they? Well, ambrosia beetles are extremely small and have established a symbiotic relationship with ambrosia fungi, which is why they're called ambrosia beetles. And my main research focuses on the Polyphacus shot-hole borer, or we'll give it the acronyms of PSHB. And this is it, this beetle right here. These are some eggs right next to it, some first and second stage larvae, and then the last one on the right, you can see like the pupae as well, and then the adult beetle. Now, you might be asking yourself, how small are we talking here? So, I have a video for you to see. Here they are on what used to be a plastic lid, and I cut out the inside of it and put metal mesh because they can actually chew through plastic. That's how strong their mandibles are. And yeah, they're pretty small. Going on to the next. So why are they considered a pest? Well, for many reasons. One is that they excavate tunnels once they found a suitable plant host. And these tunnels are called galleries and usually reside in the xylem tissue of the plant. So you might, if you can recall, xylem tissue is responsible for the transportation of water and minerals from the roots all the way up to the branches. And if that structure is compromised, you can also compromise that transportation, meaning that the branches are going to start wilting and maybe breaking off and dying. So this is the picture on your right is what it looks like on the outside. You can see the fresh grass, which is basically kind of the debris and the, let's just call debris of the beetles as they're excavating inside. And then the picture on the left is the galleries inside. Now, this is what it looks like inside of a castor bean log, and the picture on your right is an avocado log, actually. And the tunnels are there not necessarily for the beetles to feed on the plant. They're there to provide the perfect humid environment for that symbiotic fungus to grow and thrive in, because the beetles don't necessarily need the plant. They feed on the fungus. So imagine squirrels or chipmunks that have a bunch of nuts in their mouths. They have these internal pockets that they store this fungus. So as they're digging the galleries, they keep spitting fungal spores so that they can grow. And it's perfect. This is where the water resides, so the fungus grows. Now, this beetle, unlike many beetles in ambrosia in that group, this one in particular goes and attacks healthy trees instead of dead or dying trees. And some of them are oak, sycamore, willow, and most importantly, and I think what got your attention here is avocado. Yes, avocado is susceptible to being attacked to this beetle. And it can attack over 200 actually here in Southern California. And it's super hard to control, not only because of the internal structure damage of the xylem, meaning that even systemic insecticides, which is insecticide that's being absorbed by the roots of the tree, it can't transfer all over, same thing with, water. And it also mates inside of these galleries prior to dispersing. So mom lays a bunch of females and a few males who can't fly, and their sole purpose is just to mate. And so the females, once they're mated, and all the fungi is depleted from the natal gallery, meaning mom's gallery, they spread out and find another plant host, and there it is. The cycle is repeated again, which is why biological control and finding their natural enemies is the way to go, at least I believe so. My main goal is to narrow down a list of these natural enemies from the beetle's native region and then determine whether or not these potential natural enemies are safe to be released in Southern California. Because just because they can attack our beetle or this pest doesn't mean that they won't be able to attack another, say, native beetle or another native, say, caterpillar or anything of that sort. The point is to try to make sure that they only attack the pest at hand and nothing else that would damage the habitat of the environment. So this means a collection road trip, and our destination was to Taiwan. Our lab went out there and cut up some beetle-infested logs and brought them into our quarantine facility here in UCR. My task was to separate these logs into separate containers and observe and see what emerges. Our main focus was to try to find parasitoid wasps because they tend to have a very specific host range, meaning that if they attack my beetle, they are less likely to go and attack another beetle from a different group. So I spent my days observing and watching what emerges, and then I reintroduce what emerges into a beetle-infested log that I have created. I won't say created, but inducement infestation in our lab. So local beetle-infested logs, you should say. And my findings were these following parasitoids. Now, you're like, oh, they kind of look the same. So to give you kind of a better understanding, here's a video. I like to watch more than look at the pictures. So here you have the parasitoid wasp, and as soon as it finds a beetle that's about to pop out of the gallery, it engages in this pursuit to go and find it and inject an egg. And for this one, it's an adult parasitoid, meaning that it would go after adult beetles instead of their larval stage or pupal stage, which is great because the adults are the ones that are causing the most damage. And we further confirmed that these parasitoids are attacking our beetle because here you have the beetle carcasses or the dead bodies with emergence holes on their abdomen, meaning that the wasp, once it ate the insides of the beetle, eventually went and cut out a hole and then exited, only to repeat the cycle again. So yeah, this is exciting news, but there's still more work to do because we have to increase the colony size before we go on to goal number 2 in testing whether we can release them in Southern California. And that is it.
Awesome. Thank you, Dina. And thanks to everybody that's been talking about plant damage and controlling these pests. There's a lot of crazy diseases and things like that out there to keep an eye out for, especially the folks at home that might find them in their yards. I actually work on those types of pests at UCR and do things very similar to what Tobias does, but I'm not going to talk about that today. Instead, I'm going to go out of left field and talk about some of the damage, the actual kind of physical damage that arthropods can do to you, mostly to you, but also to other predators and organisms out there. I'm not going to talk about sort of the medical veterinary epidemiology of things like mosquito-borne diseases or ticks or the medical issues that you can get into. But what I will talk about is sort of the physical damage that arthropods can do to you, such as a spider that might inject venom into you. But the first thing I want to get out of the way are some semantics. And then I'll also try to dispel some myths about spiders and debunk all these crazy theories people tend to have about them. But the first thing that irks me that we'll get right today is the difference between a poison and a venom. A poison is a substance that you eat or swallow or can be absorbed through your skin that causes illness or death or injury. So, Cusco's poison. Poisons are tend to be labeled with these like skull and crossbones, and this is the typical thing you think of when you drink a poison, or the bad guy in a movie, or Snow White eats the poisoned apple. She eats it, she consumes it, and it injures her or puts her to sleep or whatever it is. A venom is a substance secreted by animals, but it's typically it's an injurious substance, and it's typically injected either by cause damage in these ways. Most of the arthropods you think of as being damaging are venomous. They're the ones that actually bite or sting you, and it hurts because they physically inject you, and then also there's a venom in there that is painful or deadly. But a lot of folks don't realize there are also a lot of poisonous insects. And we keep those terms separate. And poisonous insects are ones that if you ate them, they would cause injury to you, such as this lubber grasshopper, which is advertising to you, hey, don't eat me. This is a insect that has aposematic or warning coloration. It's bright yellows, reds, blacks, and these grasshoppers feed on a lot of noxious plants in their environment. And imbibe a lot of those chemicals in their body. So if you ate this, you would get sick. And that's what it's trying to tell you and advertise to you. Probably the most famous poisonous insect out there is the monarch butterfly. Both as caterpillars and as adults, they are poisonous because the caterpillars feed on milkweed plants, which have a lot of cardiac glycosides and heart toxins, things like that. And there's been some famous studies of birds predating on monarchs and later throwing up because they're distasteful. They're pretty gross. So they kind of learn to avoid these things. There are other poisonous insects that we find in California, blister beetles being one of those. These guys secrete a compound called cantharidin that can be pretty nasty and can cause, I'm not going to show too many gross pictures of skin damage and stuff like that, but these blister beetles can cause blisters by this process called reflex bleeding. If you grab one and squeeze it, it basically clenches its body so hard that it squirts its own blood out of its joints in its elbows and its parts of its legs, and that secretion can get on you and can cause topical injuries, blisters, things like that. So keep a lookout for those. Other ones you guys might have seen around California are scorpions. You can see obviously a stinger. They can pinch you and that might hurt, but the sting is really where they would inject venom. So this is now a venomous arthropod, not an insect, but an arthropod that can inject venom. Usually they use these to capture prey, right? But they can also be used defensively. And different arthropods use their venoms in different ways. There's a striped-tailed scorpion, another common scorpion out here. You can almost see actually a drop of venom right on the tip of their stinger right there, which are usually, venoms are usually proteins, different modifications of proteins that can be very painful or can shut down muscle functioning and things like that in something that's trying to eat them. This is another arthropod around Southern California you don't want to pick up. This is a desert centipede or a tiger centipede. And these guys actually have this modified front pair of legs up near their head. They're not actually mouth parts. They're modified legs that are hollow, and they actually pinch you and inject venom in that way. But here's a poisonous arthropod, a millipede. This is one of our common millipede that you tend to find around Southern California. And these guys, when you pick them up, they can also do like the blister beetle. They kind of secrete a lot of nasty chemicals, including benzoquinines and hydrogen cyanide, and they can give you topical blisters and things like that. So not all that fun to pick up. Another thing that's really comes to mind when people think of venomous critters are spiders, specifically black widows and a couple others that we have around Southern California that I'll show you. Black widows can be a very potent venom, but again, I want to reiterate that spiders aren't out there to kill you. Mostly what they use these venoms for are to hunt their prey and to immobilize their food, and they're just trying to kind of make a living. So don't kill spiders. Grab them in a cup and put them outside. Be careful with venomous ones, but they are very unlikely to bite you. The thing that will not bite you in Southern California, I'm sure everyone has an aunt or somebody that has, can you guys hear me? Hopefully you can hear me there. The thing that will not bite you in Southern California, at least, is a brown recluse, because we simply don't have them in the state. There have been fewer than 20 actual confirmed reportings of brown recluse in the state of California, and almost all of those have been associated with someone moving a box or something moving from the Midwest, which is where these spiders are common, and bites do occur. These are the spiders that get a moniker, the violin spiders. They get a moniker for biting you and causing this necrotic wound that can open up the skin and things like that. It's still debatable whether their venom actually causes that. More likely, it's you might scratch a bite and introduce a bacterial infection into your skin that can make the wound worse. And again, brown recluses, we simply don't have this species in California. We do have a desert recluse, and I forgot to put a map in here, but they're virtually never found in homes. So we do have violin spiders native to California, just not this one, despite what every medical expert says they're often misdiagnosed. Usually they're misdiagnosed as either this spider, which is a crevice weaver, a common crevice weaver spider, Cuculcania. These are all in everybody's homes here, but they have venom to eat to subdue their prey. It could be misconstrued with a hobo spider, another non-lethal, venomous but not lethal spider. All spiders are venomous. They all have venom, except for one family of spiders, the feather-legged orb weavers, Eulaboridae. They actually just don't have venom glands. So those ones, go ahead and pick up, no problem. But 99.9999% of spider species are totally harmless to humans, even if they did bite you, which is very unlikely. Here's the map I forgot to put in, where we have brown recluse, Lactocelles reclusa, in the red here. Totally not in California. We have one species, this desert recluse, that you'll never see unless you go to Joshua Tree and flip over a bunch of logs and look specifically for it. And that's this one down here, which also has a violin, also has 3-eye arrangement, things like that, but is of no medical consequence, really, mostly because humans just don't interact with it. Other venomous arthropods include the venomous insects you typically think of that can sting you, which are typically hymenopterans, ants, bees, and wasps. Here's a honeybee, which is well known for having a barbed stinger. It gets stuck in you. It kills itself. The worker bees that are defending the colony might sting an attacker, and they pull their guts out, basically, and they pull a venom gland out of their body that continues to pump venom into you, which kills the bee, the individual bee, but hopefully helps protect the colony from attack, for example. We do have fire ants in Southern California. They're becoming more common, unfortunately. They're an invasive species, but this is a good example of something with quite a painful sting. Most ants can sting, but they're not too painful. These ants can also bite, but they bite with their mandibles. And the ouchy part is the sting that actually has a venom that hurts. So that's another misconception to say is if you get stung by an ant, you always want to say, ouch, she stung me, not ouch, he bit me. Most ants, most hymenopterans you see are females, and the ones that sting are all females because the stinger is a modified ovipositor. It's a modified egg-laying device that only the females have, and they modify it and add a venom gland to make it a needle to inject you. And that's usually what is the most painful. In fire ants cases, they cause these nasty bumps on your skin, and they actually have a cytotoxic venom that kills a lot of your cells, which is that white pus in the middle, so not pleasant. Here's another group of organisms you probably have seen in your home. These are paper wasps, Polistes. And again, these will use their venom to hunt and also to defend their hive if you get too close to them. But these are very calm insects if you approach them correctly and calmly and you don't freak out and try to swat them. Then they will defend themselves. But you can approach them, and I've just picked nests right off of trees and moved them around, and they're very calm if you're calm approaching them. But they're so good at defending themselves that a lot of other organisms try to mimic them as well and get a free benefit from looking like a dangerous wasp. This is a Ceciad moth that looks a lot like one of those paper wasps, and it's employing Batesian mimicry to try to fool you into thinking that, hey, this is also something to avoid. I will say the last thing I'll mention about venomous, come on, I don't know why my mic is cutting out. The one thing I will say about venomous insects, the worst bite I've probably ever gotten, and this is an actual bite, not a sting, was from one of these predatory stink bugs, a podiasis, that uses a venom gland in its mouth part, in its mandible. So this is one that actually bites you and injects venom, much like a rattlesnake or something like that. And these are actually one of the most painful things I've been bitten or stung by, comparatively. So just a heads up of the various things in Southern California that can either hurt you by touching it or by stinging you. But again, to reiterate, 99+% of the critters you find out there, and the insects that we're celebrating in this insect fair are totally harmless and really cool to look at. These venomous ones are really cool to look at too, and really cool to study. Just be maybe slightly careful with picking some of these guys up. So, thanks, y'all.
Awesome. Thank you guys so much. That was a really great and really fun to learn about all the different areas of study that you guys have. So just to kind of reiterate, as mentioned in the chat, but anyone who has any questions, if you're on Zoom with us, please feel free to put them in the chat on Zoom. And also if you're watching live on Facebook, we're checking all those questions and we'll be asking them as we go now. So one of the first questions coming in, I believe, is directed towards Dina. Someone is wondering how large those parasitoid wasps are that you're studying.
Oh, they're pretty small. They're really, really small. Actually, if I can go back to my shared screen real quick. Going into this right here, the largest is this one, as I can see from the meter. Just this alone, the range is 0.5 millimeters. So that could give you an idea. And then this one, the second largest, is 0.2 millimeters for just this part of the abdomen. And then the smallest of the three is all the way on the right side. And just that head part is 0.2 millimeters as well. But that should tell you really in comparison how large or how small, but also the order of size for these wasps. So this is the largest one. Sometimes it can be up to, if it's extended, 3.3 to 3.7 millimeters in length. This one was about 2.7, 3.1 millimeters. I did kind of like a range for some of them. And then this one right here was really, really small. It was 1.8 to 2.1. It just really depends because we have different species that we're trying to separate that fall under the... Anyways, but yeah, hopefully that'll answer. Nothing like the paper wasp or anything like that, I hope.
Another question that came in was where in California scorpions are found, and if any are found in Moreno Valley.
Absolutely, they're found in Moreno Valley. You should consider yourself lucky if you live out there. You have up to maybe 3 species of scorpions. They're all fantastic, gorgeous to look at. Don't worry about them really getting into your house. They don't want anything to do with you. They do come into homes. They can be found around areas, but mostly they'll be found outside in things like brush piles or areas where boards are overlapping or stuff like that. If that is a concern, go ahead and clean up those types of brush piles and those types of areas that would encourage scorpions to live there. And that's the most effective way you can sort of keep them out of your home, is just not give them the space to find a habitat that they want to hang out in. But yeah, there are hairy scorpions and striped-tailed scorpions, and then another fat-tailed scorpion, Pseudouroctonus. So there's as many as probably 3 common ones, but maybe even more than that. The state of California, I think, has upwards of 10 or, half a dozen to 10 or so species, and they're distributed all over the state, but Moreno Valley is in a sort of desert area of inland Southern California, which is prime habitat for scorpions. At least it used to be until the houses got built there. So do keep a lookout for them. You can monitor for them with a UV flashlight. That's one really fun way to go out looking for them. And they really pop out of the substrate because they fluoresce in UV light. So if you are concerned, or if you don't want to step on one, go into your bathroom, take the UV flashlight with you, and scan your floor before walking in there. But, yeah.
Awesome, thank you. So in the beginning, there was a lot of talk about some of the diseases that are caused by insects damaging plants and kind of the damage that can come from those diseases. What is is it more likely that disease is going to damage plants, or are insects feeding on the plants kind of a higher risk? And is there a good way to kind of combat both of those, or are they separate issues that need to be dealt with?
I could take a stab at it, at least from like my the psyllid and the HLB. Because there's no cure, there's a paper that was released February of this year, and they found this stable enzyme that can actually, like, I think it could even like reverse HLB within a tree. These are all greenhouse trials, and it's going to take a couple years for that to be implemented. But until that happens, there's no known cure. So as of now, I mean, there's no way to reverse it. And once we lose citrus because they take so long to mature, it could be pretty devastating. So controlling the pest right now is essential solely for the fact of controlling the spread of this disease that has no cure.
I can go next. So I work on aphids, and they can spread viruses around. But a lot of times, aphids don't have any viruses, so their direct damage or feeding on the plant could cause more problems than the virus, especially since they reproduce by cloning themselves. So if you just have a couple aphids and it takes 7 days for them to, go to adulthood, very shortly you'll have a huge population. And the honeydew that they actually excrete, which is their poop, it's just a lot of sugar water. Mold can grow on that. So then you have aphids feeding, maybe viruses, but you have mold too. And that, all of it together, can cause a lot of problems. But as far as other herbivores, like caterpillars that chew rather than suck out sap, if you have one giant green caterpillar, the Manduka, I think they're hawkmoths when they pupate, those can eat a lot of leaf tissue. So depending on the type of herbivore damaging your plant, it may be worse to have a pathogen or better. And lastly, there is some new evidence showing that for some viruses that are typically bad, depending on the condition, like drought or frost, the plants that are infected with the virus may do better. But that's really new research, and we don't have a lot of info on it.
I can just chime in and add a little bit about psyllids in general. So unlike aphids, psyllids take quite a bit longer to reproduce because, as Tobias mentioned, they do lay eggs and go through several nymphal stages. So it can take about a month for a generation of psyllids to develop. So usually in natural settings or even maybe in your home garden, if you have psyllids, they won't build up to a population level that will cause very severe damage to your plants because there are lots of natural enemies around that will come by and eat them up. For example, ladybugs. However, in commercial agricultural settings, a lot of times we're using pesticides, and we have very unnatural conditions where we don't have a lot of plant diversity. We have just one plant growing, filling the whole field. And in those cases, insects like psyllids, there might not be a lot of natural enemies around to control them, and they can directly damage plants. But for the most part, it's the pathogens that they transmit that are going to cause more damage, I would say.
All right, then I'll chime in now. As I mentioned earlier, with ambrosia beetles, or at least with my beetle in particular, or my focus beetle in particular, the Polyphytes shot borer, it damages the vascular tissue of the plant, namely the xylem, which is responsible for water transportation. And if it's just one beetle or two on on a plant, it's fine. However, it's not necessarily fine in the long run, only because it can produce a large offspring, size of maybe 50 plus. That's just from one female beetle, and it can then usually they don't even have to fly away. They can just crawl up a couple of inches from their natal gallery and then start the infestation again. And the multitude of damage, or just the buildup of damage, both internally and also having the Fusarium fungus, which is the symbiotic fungus that they carry around, having it build up and also obstruct the water transportation does lead to the death, but it takes a long, long time, maybe 2+ years. Originally in the past, depending on the age of the plant host, if it was younger, then it would die earlier, only because it's not large enough to then utilize the other vascular tissues for the water to reach the rest of the branches. But if it has a small diameter, the likelihood chance of it dying much earlier is very possible. So yeah, here you see indirect and direct damage on the plant. So it's honestly a combination of both the physical part of the vascular tissue being compromised, along with the buildup of the actual pathogen, meaning the fungi, Fusarium fungi. So mine is it's a killer, all right.
Another question that came in is whether all aphids vector diseases or only some kinds of aphids.
that is a question I don't know the answer to. However, I do know that some viruses can only be vectored by 1 or 2 aphids, whereas other viruses can only be vectored by like 1 aphid. And that's true for the reverse. Some aphids vector or transmit not very many aphid viruses, but other aphids can transmit like up to 60 different viruses, like the generalists. Maybe I got that wrong, but maybe Jamie knows a little more too. Popcorn.
I think that was all good information, Tessa. It seems like I guess that what Tessa's struggling with is like there are so many different species of aphids, and we haven't necessarily tested in experiments whether they can each vector each different kind of virus, because there are even more viruses out there than there are aphid species. But I think in general, it seems that most aphids have the capability to transmit viruses, but that doesn't necessarily mean that they are actually carrying a virus.
Awesome. Thank you, guys. Another question coming in for Ian is why some bugs have poison or venom, and what could impact whether they have poison or venom?
That's a really good question. It definitely depends on a lot of different environmental factors. And most bugs, most insects are not really harmful or not poisonous. The ones that tend to be poisonous are ones that tend to eat noxious plants or poisonous plants, right? And they imbibe a lot of the chemicals from those plants, keep them in their tissues, and then they themselves become poisonous. And usually what's advantageous about that for those insects is that they get some measure of protection from other predators. If you taste bad, you're unlikely to be eaten by something else. However, it can be metabolically costly to be poisonous. Not many insect species can eat noxious plants and deal with the nasty chemicals. The plants themselves don't want to be eaten, right? So they produce these nasty chemicals, and relatively few insect species can overcome that and actually then use that as a weapon themselves to defend against their own predators. For example, aphids and psyllids, things like that, those aren't noxious at all, and you could eat them, or many predators like ladybird, beetles, and things like that can eat them. Venomous insects, typically there's been a lot of evolution of a stinger, of the actual venom compounds, right? And that venom is kind of a specialized weapon. And that typically happens when they either need that venom to feed on their prey, or as a defense mechanism. So that's typically what you see in bees and wasps. They have a big store of energy in their nest, and that's their whole reproductive biology, their larvae. That's a precious, precious thing for their future generations, for their evolution. So there's a lot of selective pressure for them to be very defensive, or could be very defensive of that nest, and so they've evolved venoms to sting in defense. Whereas things like scorpions and spiders, they want to sting their prey and kind of get out of the way, let it die, and they're using venom as a way to subdue their prey. And actually, most of the venoms are some modification of a digestive enzyme or protein that they inject into their prey, and it actually helps start the digestive process before they even begin feeding, which can also be advantageous there. So there's a lot of different reasons depending on the organism.
Great, thank you. This is another question for Ian. Is there a difference in damage between black and brown widows?
Yeah, there is, and there can be. Brown widow venom is typically not as toxic as black widow venom. And I should have mentioned that's another species we have in California. Actually, brown widows are starting to sort of become more common. You can tell the difference between them if you see egg sacs. Brown widows have a spiky egg sac. It looks like a sea mine, like an underwater mine. And black widows have a round egg sac. So look for those under old couches and things like that stuck outside. But yeah, black widows are the more deadly. Again, their bites may result in as much as 5% fatalities if they're not treated. Fortunately, now we have a lot of good antivenoms for them. Brown widows just are not as potent as black widows.
Awesome. Earlier, I think Tobias mentioned in his presentation that some kinds of jasmine plants can be kind of a vector potentially for the Asian citrusillid. Is that all jasmine plants? Like, would it be a bad idea to plant them in your yard, or is that a specific type that's grown in Florida?
Can you hear me? It can grow here, but it's not a common ornamental plant used in California, and that's probably one of the reasons why it devastated Florida's industry so much. And the vector pattern in Florida helped us, so we're not in the same boat they are. I don't know if maybe I could share my screen again. To my knowledge, they're specific species, so it's not all jasmine, they're just the specific ones. And there were a couple other plants as well. So there's specifically this orange jasmine plant, and it's not commonly found in California. And then there were other ones as well, right over here. And these are also offshoots of citrus. You have your kumquats, your cherry orange, and then the curry leaf is not really native here as well.
Awesome, thank you. Another question is, are there any sort of, organic insect control methods that people would recommend, especially, in urban areas, like maybe if you're, gardening on your balcony, are there specific plants you'd recommend planting or things that you could do that might help that wouldn't use as much insecticide?
I know for citrus and agriculture, controlling ant populations is a pretty big deal. There is natural biological control. I think it was Jamie that was talking about that. And the Argentine ant will actually feed on that honeydew sugary secretions, the poop from many sap-sucking pests. And they'll actually defend those pests from any natural predators that might want to prey on them. So when you have a large population of ants, tending ants like the Argentine ant, it could stop all of that biological control and really disrupt the balance. And that goes on with also saying, certain insecticides that are broad spectrum. Those will kill the beneficial insects that you want. But for homeowners, it's nice to have a diversity of plants in your yard. That way you can bring in a lot of diversity and you can harbor more beneficial insects, because some plants, they do really well at certain times of the year. So it's nice to have a variety of flowering plants.
I know for my pests, the Phylloxera herbivore, just like any emerging beetle, if they are pests, honestly, physical control, that's where it is, because insecticide doesn't really work either way, because the beetle spends the majority of its lifespan inside of the plant host itself. So early monitoring detection, if say as a homeowner with their own avocado grove, if they're going around inspecting and they see signs of infestation, it's best to just cut it off right there and nip it at the bud, than to allow the beetle population to grow in size and then affect and attack the neighboring avocado trees nearby. Same thing if, it's a lot of it is just physical control with mine, and until something more concrete is developed.
So I think that Tobias's comment about having a lot of plant diversity in your yard is a really good suggestion. The more diversity you have and the more diversity of flowering plants, you have more habitat and more food resources for natural enemies like predators and parasitoids like Dina's been talking about. But if you do have plants in your garden that are infested with certain pest insects such as aphids or psyllids, there are organic methods that you can use. For example, you can actually purchase natural enemies such as ladybugs to release in your yard. Or another good one would be lacewings. And then there are certain soaps and horticultural oils that are like plant-based natural oils that you can apply to certain plants if they're getting hit really hard with aphids, for example. And they're totally safe for the environment.
There are also biocontrol companies where you can buy predators online so that they can attack, say, aphids or other pests in your garden. So look into that. That's a great option too if you don't have the area to plant.
Awesome. Thank you guys so much. I believe our time is now up, but I want to thank the viewers for joining us today and really thank our panelists for answering all these questions so well and all the exciting research that they're doing. Yeah.